welcome back to Vanithi's weekly podcast. Today in liquid biopsy part 9, let's look at SFRP4 applications. Previous SFRP4 research has shown effectiveness in cancer in vitro cell culture, cancer stem cells and animal models. However, as a new therapeutic regimes, the clinical attempts in human circulating tumor cells and tumor spheres as a diagnostic tool are not studied to prolong cancer survival effectively, opening new diagnostic tools and market potential therapeutic avenues. The major applications for accelerating Wnt signaling drug discovery are as a new biomarker for human disease detection as CTC test, ctDNA test, promoter methylation and PCR testing. Our team have extensively studied, including my PhD findings on these aspects. For patient cell protein medicine production and treatment as viable SFRP4 CTC and sphere gene protein IDs and SFRP4 circulating tumor cells and circulating stem cells for treatment prediction. Circulating tumor cells and circulating cells gene therapies based on our lab findings to clinical trials. The isolation of stem circulating cells, tumor cells and their spheres from tears, saliva and blood sample as a total liquid biopsy comprehensive cancer care shows promise for huge commercialization potential. Of course, we need to validate on healthy volunteers and mark SFRP4 CTC range cutoff counts in normal, pre-cancerous, pre-disease condition and apply them to cancerous conditions for prevention and disease management. With the present track in the coming years for long-term goals involving global senior principal and chief research fellows, investigators, advisors, early to phase 3 trials can be conducted. The advantages of using patient CTC productions are it is E. coli and animal free, human blood cell origin will be purified using non-denaturing methodologies, it will be properly folded and biologically active with stable formulation to retain their functional activity. As a next step for direct human SFRP4 domains, that is the CRD and NLD protein production. So, what role does SFRP4 play in cancer liquid biopsy diagnostics? In CTC drug development, it can be used for stratifying cancer risk, cancer patients, measuring resistance and sensitivity, Wnt inhibitor selection treatment, cisplatin combination treatment, to check the efficacy in combination with affordable cytotoxic drug treatment for effective treatment prescription with CTC sphere dissociation, drug concentration, predicting treatment resistance and sensitivity. Manufacturing industrial scale native CTC protein productions and generating cancer IDs for Wnt inhibitor treatment selection. Based on the previous scientific evidence, we know EGFR CTC mutation detection was more sensitive in lung cancers than cell-free plasma DNA, and CTC testing was more sensitive than prostate serum antigen testing in prostate cancers. These publications bring hope to CTC testings in clinics, translating SFRP4 lab discoveries to hospitals and clinics for detecting future CTC Wnt signaling mutations and drug discoveries. Medom Therapeutics is optimistic and excited about licensing the Western Australian based STEM CTC and spheres for cancer prognosis and diagnosis as new generation SFRP4 cancer liquid biopsy diagnostics products for TGA, FDA drug development and manufacturing. Today I would like to end this podcast by leaving interesting open questions to oncologists for improving current treatment challenges with with a more intelligent option of using live cell measurement over dying cell measurement before and after therapy. Thank you.